and like that. Okay, so let's get started. So a little background. Multi-omics approaches to data analysis is becoming more and more popular, providing a better understanding of the system under study. So it's very common to work with, for example, combinations of transcriptomic, proteomic, epigenetic, metabolomic, lipidomic data. And in this webinar, we will combine transcriptomics and proteomics data and demonstrate how you can quickly identify correlations between genes and proteins analyzed on different platforms. So first we need to prepare our data set. So we start with two data sets, one transcriptomic data set and one proteomics data set, where the same samples, the same patients have been identified. And then we merge the two data sets, and we also include a column with information of if it's what type it is. Is it a gene or a protein? So here is an example of that. So here we have all the genes, and then we just added the protein data here, where the corresponding results for that sample comes in the right column. Okay, and then we are ready to go. And then we can start finding variables, genes and proteins that correlate. And here we work with Pearson correlation. So I will show you the first workflow here, where we start by loading the data set via the wizard, and then we will create the list of genes and proteins and so on. So you have all the steps here if you want to try this after the webinar. So now I move over to the software. Or before I do that, I will actually show you what the dataset looks like. So it's this one, so it's a TXT file, tab separated TXT. So here we are. And here you can see that we have all the variables. So first, initially, we have all the genes. And then if we go down, we have 150 genes or so, 167 genes. And then here we have the proteins. So in total, I think we have 300 something variables. And here are all the samples. So from 4 different treatments, 3 different drugs and 1 control group. So this is the data that we are going to use. So here I am in the software. And I just open this data set, open with wizard, I find the data set, here we are. And then we just step through the wizard. So here we have the data set again. First question is this a row count matrix? No, this is already normalized data. The separator is tab. Next question, rows are variables. So that's also correct. Next. And then we need to define the data matrix. So select the top left cell of the data matrix by clicking in it. So this is the top left cell. Next. Select the top right cell of the data matrix. So let's go all the way to the right, like that. So now we have defined what the data is. And then click on variable annotation header cell. So we have one here, and here we have the column where we have defined if this is a gene or a protein, which we will use to create lists of gene and proteins. Next, and click on sample annotation header cells, like that. So now we have helped the software to understand what is what in this file, and then we just press finish and import this data set. So here we are. So we have, let me just do like this so I can see the bottom here. So we have 72 samples and 378 variables. And right now we are looking in a sample PCA plot where we see the samples And there are some clear groups here, and they represent the different treatment groups, the drug groups, and the pink one is the control group. So before we start the analysis, we will create lists here. So we will go to the variables tab and search for type. Let's say that I would like to search for the type gene. Then I find 164 genes, okay? And this result ends up here in search. So I make a copy of this list, and this is a list of all my genes, which I will use later on. And then I do the same thing for protein, like that, and then I find 214 proteins, and in the same way I make a copy of this list. So now I have my protein list here, like that. Okay. Then I will switch to a variable PCA plot. So here I now see all my 378 variables, and I will color them so we can see which are the genes and which are the proteins, like that. And in this exercise, we will look for genes and proteins that are correlated with the gene CD74. So I will search for that. So here, so by just typing very quickly CD74, I actually find that gene. And then if I use the mark here, click on this, that one will be highlighted. So here it is, and we can use the label tool to ensure that this is the right one, like that. Okay, but there's a lot of information here, so I want to take away all the other genes for the moment and start only with that one. And the way to do that is to use the search tool again and search this time for gene protein and say that I want to search for CD74. I find one. So again, I make a copy of that. That is now CD74. Let's double click. CD74, like that. Okay, if I now make this list the input, I only see CD74. That. And then I will use the correlation tool, like that. That opens up this little dialog. And I need to click on the variable or variables. In this case, we only work with one. And then we can drag the correlation slider here. So let's say we want to find variables that have a correlation of 70%. And you can see it's a mix of genes and proteins. Proteins are pink and genes are yellow. And we can also look for negative correlation, like that. Now I probably want to make lists of these. So we create a new list, use the list tool, and just circle these genes. And they are now positively correlated with CD74. And then we do the same thing for the other one, like that. So we circle here. And here we have negative correlation with CD74. Like that. Okay, good. Next thing, maybe I would like to compare these lists in a Venn diagram. So let's open up a Venn diagram here, Venn, and then I just define what I want to look at. So I want genes and proteins, and there is no overlap there, of course. And then I also want the positively correlated and the negatively correlated. So then we can see that we have a positive correlation here of 40 genes. No, sorry, positive correlation of 28 genes and a positive correlation of 40 proteins, like that. And if I want to make a list of these, I just use the list tool again, and I click in this intersection to create a new variable list, and that is now positively correlated genes. Positively correlated genes, like that. So you can, wow, I don't know what happened with the name here, but wait, what happened here? Strange. Um, uh, Positively correlated genes, like that. So you can create a lot of lists here. And if we look here in the here, we will also see that we get in the list here, we also get the actual correlation numbers here. So we have some very high correlations. So CD74 actually has 100% correlation. And okay, so both positive and negative correlations, of course. Good, and then you can export this list to have the information here. Okay, so that was an example of the first workflow. You can also export a correlation matrix where you get all the correlation numbers in one big file. So in the file menu, File, Export, Variable Covariances and Correlations here. Let's just create a report that is called correlation. And then here we need to select which annotation we want. So we want the gene protein and we want the correlation, and okay. And if we go to the desktop here, we will find the correlation report, I hope. Maybe that was not where I put it. So let's just do it again. So I struggle a little bit to find my mouse here. So export here, correlation. Yeah. Okay, so let's put it on the desktop instead. It's easier to find. Correlation, like that. Save. Again, gene protein, correlation. Okay. Here it is. And here you have the full report of all the combinations of variables here and the correlation numbers. Let's continue. Let me just check if there are any questions here. Chat. Nope, good. Okay, so now we are going to move over and also include a statistical test, because just because variables are correlated, it does not necessarily mean that it's statistically significant. So we are going to use linear regression, and linear regression you can use when you have numerical sample annotations like time, dose, and age to find variables that either increase or decrease over the annotation. And in this case, you can use some of your variables as sample annotations. And the variables, of course, have numerical values. And then you can use linear regression to find statistically significant correlations. So we need to prepare our data a little bit again before we start. So you need to edit the input file so that the gene and protein names, are in the same column as the sample annotation labels. So everything needs to be in the same column. And then we import the data set using the wizard, and here then we will treat, in this example, all genes as sample annotations. So let's have a look at this data set as well. So here we have it. This is the one that we're going to use. And here you can see that now the sample ID treatment, these annotations are in the same column as all the gene names, which we are going to treat as sample annotations. So that's a prerequisite for that to work. Okay. Close down, and here we are again. So now we will just close down this data set, discard, and open up the other data set. So we can also close this correlation window. So File, open with Wizard, find this other data set, this one, and then we enter it. So here you see the same file. No, it's not a row count matrix. The separator is still tab. The rows are variables. And now the next, select the top left cell of the data matrix. Okay, since we now are going to treat all the genes as sample annotations, the data matrix does not start until we come down to the proteins. So this is the first value of the data matrix here, the first protein here. Next. And then we want to go all the way to the right and find row 45, I think. So, okay, here. So now we mark the data. Next. Variable annotation header cells. We don't really have a good header cell, so when we don't have that, we just click on the first variable cell, like that. Next, click on sample annotation header cells. So this is a sample annotation, this, and this is also a sample annotation. And if I go all the way down here to the last gene here, and then I press Shift, and then press this cell, then it grabs everything. Okay, so now we have defined the file again, and we press Finish. And that opens up the data again, data set in a PCA plot. So again, we can color it on treatment, like that. Color. Okay, and in this exercise, I will actually just switch directly to a heat map, and we will maximize the heat map, and we will hierarchically cluster it. And yeah, before I go there, what we are going to do here now is that we're going to start by analyzing the correlation with, or do a linear regression on the gene ABCC1. So if I color by that, it does not make sense at all, because each result has its own color. So what I first want to do is to create a color gradient. So I do that using this little icon here and say color arrange as a gradient. Okay. Select the first value. This is the first value, and I actually want to start on yellow. So I start by changing the color and then press OK. So that's the first value, and then I want to go to the last value in the range. It's green. I'm okay with that. And the values must be sorted in the right order. If they are not, you can move them up and down using the arrows here. So I select this one. Okay. And now I have my color gradient. So yellow is a low expression value and green is a higher expression value. So now I don't want to do a hierarchical clustering here. Instead, I want to sort or order my heat map on this the values for this annotation. So it's not now ordered for the expression of ABCC1. Okay, and then we are going to do the actual statistical test. So we want to do a linear regression on ABCC1 to find proteins that have a that either well, increased or decreased over the annotation. So if we choose a really good Q value here of 0.01, we end up with 138 proteins meeting this criteria. And if I zoom in a little bit, you will see that I can see the protein names here. Okay, so that's and of course then I can export this list that then contains all the proteins that are have that has a good Q value for the regression for ABCC1. Okay, so we can go through all of the genes individually and see where we have good correlations with which proteins, but that's quite a tedious work. So instead, we will move to the biomarker workbench, because here we can analyze all the genes, all the sample annotations in batch mode and get a quick overview and identify the strongest signals. So we will open up the biomarker workbench, which is this little icon here. And we add all the annotations like this. So you can see we added everything. So let's also maximize here. Okay, so and the software automatically assigns a test based on the data. So all the genes, the suggestion is to use linear regression, and for the treatment annotation and multigroup ANOVA. And you can change that because you have all the most of the tests here available. Okay, and then we just press run, and immediately everything is calculated and we get the results here. So here we can see how many proteins have a Q value of less than 0.05 for this gene, this gene, this gene, and this gene, like that. And we can sort on that as well. Okay, and then we also have the detailed view here, like that, where we can add the R statistic, because when you do linear regression, the R statistic is actually the correlation value. So here for each of the genes, we have the statistical values, the P and Q values. We can actually take away the P value so that we don't have that many columns here. So we have the Q value and we have the correlation value, the R value, which we can sort on. And we can see that the best genes here are also, the best proteins here also have good correlation values. And we can sort on that one. So you can sort on any column. And here we have the results for all the genes and also the treatment annotation. And then we can export. So we can create variable lists from here by just marking which proteins we are interested in and just export that list back to our main window. And we can also export the full table and then work with that in Excel and so on and get the full overview of all of the results. So it's a really powerful tool, the Biomarker Workbench. And here are some more details on how you can do what you can do. Okay, and finally, now we have generated a number of results, and now we would like to visualize these results. And let's start by visualizing them in a scatter plot. So let's just do like this. So let's change this to a scatter plot, a scatter sample. And here I would like to have here a sample annotation on the x-axis, and the sample annotation that I want here is this gene. I can pick anyone, and in this case I want this gene. And I want a numerical scale. Otherwise you won't get the correlation coefficient in the plot and the correlation line. So you probably want to do that. And then I want to select a couple of proteins here. So I go to the variables tab and I add the Q value column and sort on that, so I get the best ones on top. And then I add maybe the four best proteins. You can see that we have a positive correlation here for these, and here we have a negative correlation. And you can see the correlation coefficient here, which is the same as the R value. And we can color here. We can, of course, color on the this annotation if we want, but that's actually what's on the x-axis. So let's color by something else. So let's color by treatment, for instance. And then, so if we maybe change color on that, like that. Yeah, so here you also get an idea of how the different treatments function here. Okay, and if you wanted, so now we have the protein name here, which is hard to read. So instead we can say that we would like, for the y-axis, we would like the label to be on top. So now you see the protein names here, like that. Good. And another plot that is quite good to use is the line plot. So let's pull up that one, line sample. Yeah. And here we can have the active samples on the x-axis. We start by that, and then we pick the same variables again and have a look at them in a line plot like this. And here we want to make sure that we have, okay, let's go back one step. So now we have sample ID on the x-axis. We probably would like to have expression value for our gene of interest here. So let's go back and pick that instead. Then we see that we have a positive correlation for 3 of the genes and a negative correlation for 1 of them. Yes, I think that was what I intended to show you. So 2 or even 3 interesting workflows, if we include the biomarker workbench, for how you can work with multi-omics data looking for correlations. So by that, I now open up for questions. I open up the chat if you have any questions. So don't be shy. So maybe this was crystal clear. In that case, as I will send you the slides so you can try this on your own. And while you are thinking, if you have any questions, we can have a look at other upcoming webinars. So next week we will introduce our new cloud solution, which means that you can run Omics Explorer as a cloud service. And then we will do basic trainings, at least 2 before summer, and there will probably be 1 more webinar which has not been defined yet here in the list soon. Okay, I got 1 comment that it was crystal clear. Okay, good. Perfect. So okay, I got a question here. What should I do if I don't have a TXT file and start my analysis from 10x data files where proteins or genes are in the same variable list? Yeah, I think what you need to do is then maybe to, if you can introduce a column, I don't know if you can, of course you can import a variable annotation which tells us that which of the variables are genes and which are proteins. So then you get that type into your data set, and then you can do exactly the same as we did here, where you actually see which are the genes, which are the proteins, and so on. I think that should work. And if you want, so if you have this external annotation file where you define if it's a gene or a protein, you can then just go here, import variable annotations, and import that file, because then it will recognize the unique ID, which is the gene or the protein name, and then it will add the annotation gene or protein to that. Okay. That should work. And finally, if you come up with more questions, you are very welcome to contact us, either me, or if you're already in contact with another salesperson, you can contact them and or support. So by that, I would like to thank you very much for your attention and have a good rest of the day. Bye bye.